Hello and first welcome Discount Seth Rogen and Doffy for being first. And I will get to your questions in a second, Seth Rogen. And also welcome Bob Marley. Always super appreciated to have anyone pass by, but especially you folks. But yeah, I am back, and I think this is the third day in a row of streaming, and I plan to stream again tomorrow. Although maybe I'm entertaining the idea of like a gaming stream or something more light or less prepared. Like today, I put in a decent amount of effort of preparing the content. Because like, yeah, the paper that we're going to cover and do a deep dive on, it will likely involve me drawing things out more on my now tablet that I own. We're going to see how that goes. I did a trial run of that about 2 weeks ago. I think it went well. At the very least, I got some messages containing some positive feedback that was very heartwarming and great to read. So that just gives me a little bit more confidence that what I'm doing is not a complete failure. But your question, Discount Seth Rogen, so which are fantastic questions. So in terms of dopamine being synthetically synthesized, I would say, so I haven't looked deeply into it, but almost like virtually close to 100% certainty that dopamine has been synthetically synthesized because like it's not a super complicated molecule. And well, if I remember correctly, I'm even gonna like look up the structure now. Might as well just get right into it. Because like it's just like synthesized from some amino acid precursor. Yeah, it's a very simple. Synthesizing this is super easy. And we have, yeah, L-Dopa, which we often give this as an actual treatment for Parkinson's, because I believe it's a precursor for a dopamine. So virtually no doubt if we can make the precursor, or one of the precursors for dopamine, dopamine itself, like no problem whatsoever. Like a very, this is probably like elementary organic chemistry. I'm sure if I like dig through my notes of organic chemistry, I can probably find the way to synthesize it. And then to your second question, can dopamine pass through the blood-brain barrier, which is a fantastic question that I'm a little bit less certain about. It's a small molecule, as in, well, yeah, it's small. I lean towards, well, actually, what is the metabolism of L-Dopa? Since do go into the pharmacokinetics and dynamics. I lean towards no. I don't think there's any active transported. Let's see. Yeah. I wouldn't think so. But yeah, levodopa, yeah, that actually makes sense. This does, and then probably when it's in the brain, converted to dopamine. But even then, yeah, okay. Well, it seems like the answer is pretty. The answer would be no. But even if dopamine gummies like were a thing, and it does cross the blood-brain barrier, I think it's a little bit less certain whether that would actually produce some sort of reward. Because I'm not a neuroscientist, but what I understand, and you know, actually from actual conversations with neuroscientists, dopamine, in and of itself, I don't think generates like pleasure. Largely, I believe, involved in like reward seeking. Yeah, I don't think you'd get any sensations really, even if dopamine crossed the blood-brain barrier. In that, the pleasure would come from like the gummies itself, and like whatever, probably the sweetness associated with it, as opposed to the dopamine. but it's totally a fair question and a good question to ask. I wonder what are the long-term environmental and ecological consequences of using artificial embryos first. So that's an excellent question. Also, that I guess we're gonna dive right in right from the get-go. But I guess a little bit before, well, obviously I'm back, and I actually got a surprising amount of sleep last night. I think my caffeine abstinence is paying off. Day 12, logging days being caffeine free, sort of like in rehab, so to speak. And yeah, last night I think I got almost like a full 8 hours of sleep, which I don't think that has happened for quite a while. Almost to the point of like, so too much sleep, that it just makes me I wake up even more tired somehow. There's like that sweet spot of too little is bad, too much is also bad for me. But either way, not gonna complain. I definitely prefer getting more rather than less. So I think I'm gonna stick, at least for now, for the time being, caffeine free. Much as I don't want to, as much as the temptation is for every morning to, make my caffeine drink, basically pre-workout. How much is it the desire for caffeine, or how much is it just I'm so habituated to my morning routine that like I almost have to like catch myself or make sure that I don't take my pre-workout, just so habituated to the routine. I think that's sounds silly to say, but I think that's one of the things when it comes to recovering from any sort of like substance use disorder. Not that I think I had. I don't think you can have an official substance use disorder with caffeine. I think a large part of it is just like whatever, breaking the routine and breaking the habit. So yeah, all that to say, if things continue on the trajectory, I also should be back tomorrow, assuming I get another good night's sleep tonight. I'm decently confident I will. And then hopefully next week, again, still trying to aim for that, 3-4 streams per week. And I'm probably gonna post my schedule for next week in the Discord on Monday or Tuesday. And yeah, if anyone wants to join the Discord, definitely more than welcome. Great resources. And you can also suggest topics and request the studies, although I should start posting the studies that I cover on stream. So yeah, on this stream, we're going to go through this briefly. So this is an article, obviously a BBC article, so it's not like an original study, but it is referring to an original study that I do have, but it's a preprint, so it's pretty much covering this study. Accelerated article preview. It was published, about a month ago, early September. And so, accelerated article preview, so, it's still basically unformatted. I think by now it's been fully published, probably, and it's a better format, but I can only get access to the largely unformatted. Not going to go through the whole thing because it's quite long, understandably, because it's a pretty ambitious endeavor and research to carry out, and two, because I'm definitely not qualified to dissect everything that they're doing here, and I definitely would have to do a lot of other research to understand everything that they're doing here, because there's definitely a bunch of things, quite a few things that I don't understand what they're doing. Not my area of expertise. And even some of the figures, I definitely can't interpret every single figure. Because again, what they're doing is understandably quite complicated. So, all that to say, there's this mainstream media article referring to it. And then, the other study that we're going to do a deep dive on, I think, is a nice combination of, biology / biochemistry / medicine, synthetic biology. It also relates to my own condition. because it really is, at least the ultimate goal of the research, is to develop, basically a new and better way to do patient monitoring for patients like me that have my condition, like specifically Crohn's disease or IBD patients, and like engineering bacteria, so that they can actually maybe do some like real-time monitoring of a certain biomarker that's relevant. And it's, I think, super cool research. So, scientists have grown an entity that closely resembles an early human embryo without using sperm, eggs, or a womb. And this, I think it was just like yesterday or 2 days ago that I covered the research where, the artificial womb that sort of appears to be on the trajectory of being approved for starting human trials. As of right now, the artificial womb only tested on non-human animals, think like lambs, maybe pigs. And then now it seems like it's potentially on the verge of starting human trials for an artificial womb. Like, two things are sort of like working in parallel. Oh, thank you so much for the lurk, Discount Seth Rogen, much appreciated. And also, thank you for the great questions. Those were absolutely great questions that I absolutely had to think about. But yeah, you have, two research paths working in parallel. The generation of, an artificial embryo, and then the generation of an artificial womb. And that, I don't know how long it's going to take to like fully develop these to where they can actually be used for, fully human purposes. Probably like the artificial womb is like, whatever, decades away, probably at least 10, 20, maybe 30 years. And this, I have no idea. I speculate. Like that's also maybe 10, 20, 30 years, but it seems like maybe there's the potential at some point that we can pretty much create an artificial embryo and then, grow and development using an artificial womb. It is crazy and weird to think about. So, the Weizmann Institute team say their embryo model, made using stem cells, looks like a textbook example of a real 14-day-old embryo. It even released hormones that turned a pregnancy test positive in the lab. The ambition for embryo models is to provide an ethical way of understanding the earliest moments of our lives. The first weeks after sperm fertilizes an egg is a period of dramatic change, from a collection of indistinct cells to something that eventually becomes recognizable on a baby skin. This crucial time is a major source of miscarriage and birth defects, but poorly understood. It's a black box, and that's not a cliche. Our knowledge is very limited, Professor Jacob Hanna from the Weizmann Institute of Science tells me. Embryo research is legally, ethically, and technically fraught, but there is now a rapidly developing field mimicking natural embryo development. This research, published in the journal Nature, is described by the Israeli team as the first complete embryo model for mimicking all the key structures that emerge in the early embryo. This is really a textbook image of a human day 14 embryo, Professor Hanna says, which hasn't been done before. Instead of a sperm and egg, the starting material was naive stem cells, which were reprogrammed to gain the potential to become any type of tissue in the body. So, yeah, it seems like their strategy, at least based off of what I'm understanding here, is like they took stem cells and essentially like rewound the clock, so to speak. Pretty much trying to, going to time zero, the beginning, I guess. I don't know if they actually got to, just one individual cell. So basically either that or something close to that, and letting it develop for 14 days. So chemicals were then used to coax these stem cells into becoming 4 types of cell found in the earliest stages of the human embryo. Epiblast cells, which become the embryo proper, or fetus. Trophoblast cells, which become the placenta. Hypoblast cells, which become the supportive yolk sac. And extra embryonic mesoderm cells. A total of 120 of these cells were mixed in a precise ratio, and then the scientists step back and watch. So creating a model, yeah. Here are the 4 types of cells. Yeah, you have the stem cell. Oh yeah. They mix. So yeah, they start off with 120 cells, mixed and grown in shaker, and then I don't know if it's actually spontaneous, but at least the way that they're framing it here, it spontaneously forms embryo-like structure. About 1% of the mixture began the journey of spontaneously assembling themselves into a structure that resembles, but is not identical to, an embryo. I give great credit to the cells. You have to bring the right mix and have the right environment, and it just takes off, Professor Hanna says. That's an amazing phenomenon. The embryo models were allowed to grow and develop until they were comparable to an embryo 14 days after fertilization. In many countries, this is the legal cutoff for normal embryo research. So it seems like specifically, cut off the time to development for, understandable, ethical and moral considerations. So presumably, maybe, it could have developed further, but, just not given enough time, understandably. So despite the late night video call, I can hear the passionate Professor Hanna gives me a 3D tour of the exquisitely fine architecture of the embryo model. I can see the trophoblast, which would normally become the placenta, developing the embryo, and it includes the cavities called lacuna that fill with the mother's blood to transfer nutrients to the baby. There is a yolk sac, which has some of the roles of the liver and kidneys, and a bilaminar embryonic disc, key hallmarks of this stage of embryo development. The hope is embryo models can help scientists explain how different types of cell emerge, witness the earliest steps in building the body's organs, or understand inherited or genetic diseases. Already, this study shows other parts of the embryo will not form unless the early placenta cells can surround it. Bill, welcome. And YouTube chat, yes. Shout out to you. Out there, alone in the void, the YouTube chat. But you gotta start somewhere. But hope you're doing well. Great to see you. There's even talk of improving in vitro fertilization success rates by helping to understand why some embryos fail, or using the models to test whether medicines are safe during pregnancy. Doesn't seem like the goal is to create a synthetic life, at least not now, but largely to actually have some sort of model that we can use for future research to understand, early stage development, and as they say, maybe as a testing ground to see, which therapies or medicines are potentially toxic or not to an early embryo. Professor Robin Lavelle Badge, who researches embryo development at the Francis Crick Institute, tells me these embryo models do look pretty good and do look pretty normal. I think it's good. I think it's done very well. It's all making sense, and I'm pretty impressed with it, he says. But the current 99% failure rate would be proved, he adds. It would be hard to understand what was going wrong in miscarriage or infertility if the model failed to assemble itself most of the time. I'm glad to hear that. I'm glad you tried it. Yeah, because totally not for everybody. And totally, as I tried to warn on the Discord, it's very heavy. And typically, like I said, use a lot of Parmesan cheese. Not everyone likes Parmesan cheese. I happen to like it a lot. That's one of the few dishes that I'll make from scratch. I only consume it a couple of times a year because it's so freaking heavy and calorie dense, as it's pretty much butter, cream, and cheese. Pretty much the primary constituents. It's definitely an indulgence for me, for sure. And when I make it from scratch, I'm using almost a full block of Parmesan cheese. It's a little bit ridiculous, or comedic. So I've only heard of ziti. I don't think I've ever consumed. I believe that's I'm not even gonna... I've heard of baked ziti. Yeah, baked ziti. So all that to say, don't think I've ever had. It looks good. And I'm sure there's a vegetarian version of it. But let's just say. Even when I wasn't vegetarian, this is probably something that I would like. And like, yeah, fettuccine alfredo is like one of the few dishes that, let's say, fully prevents me from going vegan. Oh, and XROX Fist, welcome. Thanks so much for passing by. And tortellini is great too. Cheese tortellini, definitely a favorite of mine. Yeah, I mean, I love a lot of pasta dishes. And like, yeah, and even though there are vegan versions of fettuccine alfredo, you know, not quite the same as, you know, full dairy and cheese fettuccine. And like, I'm not gonna lie, there's a little bit of guilt when I consume it that sets in, which is why, well, in addition to it not being super healthy for you to consume every single day, which is why I don't. It's like, yeah, a combination of like, I can't afford to eat fettuccine alfredo every day. Not only just the finances, my health, and like, yeah, I do want to limit my dairy consumption, even though I don't consume that much. But still, you know, I'm in the direction of no dairy. I'm like, yeah, that's pretty much like a little dairy that I consume. That fettuccine alfredo that I make, like, you know, 2 or 3 times a year. So, yeah, the work also raises the question of whether embryo development could be mimicked past the 14-day stage, right? Seems like they intentionally cut off at the 14-day stage for understandable reasons. This would not be illegal, even in the UK, as embryo models are legally distinct from embryos. Interesting. Right. And I think this introduces some potential ethical questions for people. So, you know, without you know, getting into a debate about abortion, but, you know, some people, their position is rooted, or rather, the position about where personhood begins is rooted in, you know, implantation, you know, aligning of the uterus. Okay. I don't think that's a good line, but some people have, that's where they draw their line in the sand, so to speak, of where personhood begins. And so it's like, you know, what do we, you know, what does this person do? Or like, is the artificial embryo that has never touched uterine wall that not a person? As in, like, can you make a synthetic embryo here, assuming it gets better? And let's say it develops to, you know, whatever, 20 days, 30 days, a couple of months. So like, it never becomes a person because it never implanted in a uterus. But yeah, that poses a challenge for those individuals that take that position. I agree. It is a poor, that's why I think, you know, technology is like, yeah, it tends to push people's, you know, ethical positions, or put them in very difficult moral and ethical position. And I think if this pretty much comes to fruition and gets further developed, which there's every reason to believe that it will, then like, what do those people do? I took some friends to a Bosnian restaurant, hoping for them to try some new food. Gave them some nice recommendations. They got fettuccine alfredo, not very I'm not gonna lie, I am completely ignorant on Bosnian cuisine. Yeah, I'm not even gonna begin to guess what it's largely consists. But I mean, I can't blame anybody for going to any restaurant and ordering the fettuccine. So yeah, you know, in like a few years, or like 5-10 years, when this is more developed, what is Ben Shapiro gonna do? Of like, you know, we now have the power for artificial embryos and artificial wombs. So like, where's the personhood line for that individual? Oh, and DZ Drunk, welcome. Always great to see. I hope you're doing well. What about human chimeras? Haven't artificial embryos have already existed? So chimeras are already basically a thing. There's actually, I believe, a couple of examples of human chimeras. Case reports. Not very common. But I mean, well, so, you might be referring to chimera differently than, you know, how it's referred to in, let's say, you know, biology. But yeah, chimerism is, you know, not super, oh, yeah, not super special, or like, it already is a thing. But yeah, sort of the human-animal hybrid, that might be a different thing. That is probably more what you're referring to. But there's totally genetic chimerism. That is, let's say, a thing. There's, as they're highlighting here, there are case reports of chimera. But no, I don't think artificial embryos have already existed. I mean, artificial in the sense that, like, we can create embryos in the lab, you know, in vitro fertilization, those artificial embryos. But this is artificial embryos in the absence of sperm or egg. Okay, this is like pretty much entirely synthetic. Human embryo without sperm or egg. This is taking a stem cell and basically like rewinding the clock, so to speak, to put it like very crudely. They're doing something way more sophisticated than that. But that is sort of fundamentally what they're doing. And yeah, I'm not even going to speculate on Kazakh cuisine. I'm probably the last person to speculate on any sort of cuisine. My culinary tastes are not that sophisticated. Most of my diet consists of smoothies and protein shakes, and like whatever, baby carrots and cherry tomatoes, and super boring. So where was... Yes. Here. So yeah, some will welcome this, but others won't like it, Professor Lovell Badge says. And the closer these models come to an actual embryo, the more ethical questions they raise. Totally true. They are not normal human embryos, they're embryo models, but they're very close to. So should you regulate them in the same way as a normal human embryo, or can you be a bit more relaxed about how they're treated? Professor Alfonso Martinez Arias from the Department of Experimental and Health Sciences at Pompey Fraude University said it was a most important piece of research. Work has, for the first time, achieved a faithful construction of the complete structure of a human embryo from stem cells in the lab, thus opening the door for studies of the events that lead to the formation of the human body plan. Researchers stress it would be unethical, illegal, and actually impossible to achieve a pregnancy using embryo models. Assembling the 120 cells together goes beyond the point an embryo could successfully implant into the lining of the womb. So yeah, so totally not viable now, but that is just like for now. Whether we actually get to it being viable, open question. But as of now, it's not an actual full embryo. I guess maybe a pseudo embryo that is not viable. But let's just say, have little doubt that we're going to get to that place, like a fully artificial embryo, that likely, which is scary to think about. So yeah, this is the actual study that article is referring to. Like I said, not going to read the whole thing. Maybe at some point we'll dive deeper into it, of the things that I can understand. And yeah, a whole host of researchers from across the world involved here, largely out of Israel. But you have Sweden, Belgium, you have Canada, even, well, I know this university. I've been, who is it affiliated? This person here? I might be able to reach out to this, or like track them down. So just the abstract, basically. So The ability to study human post-implantation development remains limited due to ethical and technical challenges associated with intrauterine development after implantation. Embryo-like models with spatially organized morphogenesis of all defining embryonic and extra-embryonic tissues, post-implantation human conceptus, that is embryonic disc, bilaminar disc, blah, 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 remain lacking. So, mouse-naive embryonic stem cells have recently been shown to give rise to embryonic and extra-embryonic stem cells, capable of self-assembling into post-gastrulation mouse structured stem cell-based embryo models, blah, blah, blah. All this to say, this is just like early development. Here we extend these findings to humans, while using only genetically unmodified human-naive embryonic stem cells. So yeah, or in HENSM condition. Such human fully integrated spatially, how does it stand for? Spatially organized morphogenesis. Recapitulate the organization of nearly all known lineages and compartments of post-implantation human embryos, including epiblasts, hypoblasts, yeah, pretty much those 4 tissues. These human complete SCMs demonstrated developmental growth dynamics that resemble key hallmarks of post-implantation stage embryogenesis up to 13 to 14 days post-fertilization. This includes embryonic disc, bilaminar disc formation. Yes. This is maybe, so basically just the BBC article all over again, just technical jargon. So, and I believe there are multiple research groups working on this, because I came across another paper that I do actually have full access. This one. So this was published earlier this year, summer. Totally different research group. I believe. Can't remember where it's out of. Not that it matters all that much. This is costly. But yeah, this is also stem cell derived model of the post-implantation human embryo. But from what I understand, like it's fundamentally like all aiming for like the same thing. Sort of like trying to get as close to an embryo or an actual embryo as possible using stem cells. I believe this paper refers to this paper and says like basically that they do it better. But this is also still published in Nature, one of the most prestigious journals, if not the most prestigious journals, according to some people. But yeah, this is out of Cambridge, University of Washington School of Medicine, etc. So, and California Institute of Technology. Still, a whole host of institutions involved, and I have no doubt that so highly technical research being carried out. Yeah, definitely cannot dissect. It would take me hours of independent research to dissect. So, with that, moving on to pretty much topic and the main piece of research that I want to dive into, that we are going to dive into, is this paper. Okay, like I said, it's pretty much good example of biology, biochemistry, medicine, synthetic biology, coming together to carry out some cool research, or like at the intersection of all these fields. And it's engineered calprotectin sensing probiotics for IBD surveillance in humans. This is just like fancy way of saying genetically modifying bacteria to monitor a certain type of patient, IBD patient, and to monitor something specific, a specific biomarker that is relevant for these patients. And I've covered research similar to this in the past. I specifically covered this paper, so this is also engineered bacteria to detect tumor DNA. I pretty much went through this entire study. Super cool. And so it's actually fairly similar to what we're going to go through. But there's definitely some distinct differences. But this is detecting tumor DNA, but in this case, they were, as proof of concept, they were using colon cancer as their model. And so, like, they were engineering DNA, basically in the gut or, in the gastrointestinal tract, and then to detect for potential colon cancer. And it's roughly the same principle here of using a bacteria with the intention of like, hey, there's bacteria that will reside in your gastrointestinal tract, and then you can monitor things through basically your waste, the stool samples that these patients would give. And pretty much the overall same principle applied with this research, where they were also stool samples, but they started, I believe, with mice. Well, they started in vitro, then they went up to like an organoid, like and then they've, I think, fully moved up to actual mice, if I remember. But either way, I mostly just want to get across that very, very similar. And so this research was published a few months ago, summer, and they pretty much take the overall same approach. Like I said, they first start off sort of in vitro, proof of concept of like, hey, does this even work before we actually try it out in patients, in an actual animal, and then they sort of like systematically scale up, going from in vitro to, I would say, in vivo adjacent. Not fully using it in patients, but showing that pretty much like this has a very real potential of being used in actual human patients. So they're like in vivo adjacent. So bacteria in the gut microbiome have evolved complex mechanisms of sensing and responding to external stimuli from the host. Over the past decade, advancements in molecular biology and nucleic acid sequencing have led to the emergence of synthetic biology as a potential tool to develop novel, non-cell-based and cell-based clinical diagnostics. That synthetic biology, the way I describe it, is just a combination of engineering and biology, or like the interface between engineering and biology. Advancements in synthetic biology have demonstrated that cell-based biosensors can be engineered by placing a reporter gene under the control of a promoter that is sensitive to the presence of specific environmental stimuli or biomarkers. Several microbes have been designed previously to serve as potential diagnostics to human infectious diseases and cancer. So what does this actually mean? I'm going to like expand upon a little bit later. But yeah, pretty much, engineering bacteria such that they respond to something of relevance, and like when they detect it or sense it, like it sends off like a signal, an easily measurable signal. Several microbes have been designed previously to serve as potential diagnostics to human infectious diseases and cancers. Furthermore, past studies have demonstrated that recombinant probiotics can be used for successful detection of gut inflammation, both in vitro and in murine models of IBD, just fancy terminology for mouse models of IBD. However, these models either lack specificity to gut inflammation or detected molecules that have not been demonstrated to be correlated with inflammatory bowel disease activity in humans. Right, so they're pretty much setting the stage or describing past research where it's like, similar results have been obtained using similar strategies, but like the engineered bacteria that were used in the past or engineered in the past, they were basically not applicable to IBD patients, where it's like, they may have engineered a bacteria that can detect inflammation, but not necessarily the inflammation associated with IBD or like Crohn's disease or ulcerative colitis. So it's not specific for that condition. Because as they highlight, lack specificity to gut inflammation or detecting molecules that pretty much don't have much relevance to inflammatory bowel disease. So they're looking for something that is like specific to this condition that will basically allow you to, ideally, if this like actually gets up to full human scale, it's like an actual doctor, or in this case a gastroenterologist, can like use the information to make clinical decisions, because the biomarker, okay, that use here specific to inflammation in the gut, and it's like one of, as of now, like one of the best sort of characterized and understood, of like it correlates well with the inflammation that is occurring in these patients, like myself, where it's basically, the higher the biomarker, the more the inflammation, and that if the biomarker is, trending downwards, there is a high degree of confidence that the inflammation is also trending downward. So IBD is a spectrum of chronic autoimmune inflammatory disorders of the gastrointestinal tract, ranging from ulcerative colitis to Crohn's disease. So I think this is a little bit questionable, in my opinion. Globally, the prevalence of IBD has been steadily increasing from 3.7 million in 1990 to 6.8 million in 2017, particularly in regions with historically low rates and more limited health care resources. This is like a known phenomenon, and that generally speaking, IBD is thought of as a condition or disease of the developed world. But it seems like as countries become more developed, like rates of IBD, it does increase. And it was thought at some point, way back in the day, that this was largely due to like increased diagnosis, or rather detection and diagnosis. Because diagnosing it, or rather detecting and diagnosing it, is not the easy thing. Rather, it takes a little bit more sophisticated, medical equipment. Actual colonoscopy is the gold standard, and so basically, the type of resources required to detect and diagnose it not readily available as much in developing countries. And so it was thought, well, yeah, as countries develop and they get more technologically advanced, that they're just in a better position to detect and diagnose it. But at least based off of what I've read of the most recent data, that doesn't explain the phenomenon enough, where it's not just an increase in detection and diagnosis. Appears to be, at least from what they're gathering so far, something appears to be happening either in the environment or genetically or both, that it appears to be resulting in increased diagnosis in these. But IBD is characterized by a progressive, chronic, relapsing, and remitting course of diarrhea, rectal bleeding, and abdominal pain that can lead to irreversible bowel damage and colorectal cancer if relapses are not identified early. Since the clinical course of IBD varies from patient to patient, the optimal method for monitoring disease activity is difficult. Previous studies have shown that patient-reported symptoms are poor predictors of disease activity. Right. And this is one thing I think is, at least for individuals that have it, is important to keep in mind of your subjective symptoms and what you experience subjectively as a patient, not necessarily an accurate reflection of what's going on physiologically in terms of the inflammation that's occurring either in the GI tract or elsewhere. There can be a little bit of a disconnect where you can have severe symptoms and the patient can be experiencing severe symptoms, if not disabling symptoms. But then when we go and actually assess their inflammation, it's mild to moderate inflammation. And then there's also sort of the flip side of that, of the patient can almost have mild symptoms, or they'll report feeling generally okay, or not feeling that they're having severe symptoms. And then when we actually go in, the patient is off the charts, so to speak. Basically, we don't want to rely on what the patient reports as their symptoms alone. And generally speaking, they want to go by objective markers And the objective marker that they choose here is like one of the best characterized. And yeah, as they say, the current gold standard of disease surveillance through colonoscopy is invasive, costly, and requires medical expertise. And C-reactive protein, another biomarker, typically assessed through a blood test, is a systemic biomarker that indirectly quantifies disease activity, but it is non-specific to IBD as it is elevated in other infectious and autoimmune illnesses. This is just a generalized marker for inflammation. So it's not, like, yeah, it'll be elevated. It'll be very often, not always, but often elevated in IBD patients, but it will also be elevated in other patients with inflammation. So it's not specific to the inflammation that is IBD patient. So calprotectin, a stool biomarker that is both sensitive and specific to gut inflammation, is currently the clinical laboratory gold standard for IBD disease activity surveillance, but results usually take 1 to 2 weeks to obtain in the clinical setting. Thus, a clinical gap remains in methods of precise, efficient, non-invasive monitoring of disease activity. So, yeah. The biomarker that they've highlighted, this calprotectin, this appears to be like specific to the inflammation that is seen in IBD. That, pretty much derived from stool samples, because so the way it is generated, it is contained in neutrophils, and neutrophils are basically one of the cells of the immune system. Yeah, they're a type of a white blood cell. Okay, and so they play a part in the inflammation that is observed in IBD patients. Okay, and so it's like, when you have high inflammation, okay, then you have this high concentration of neutrophils, and then yeah, the neutrophils, like, they produce this calprotectin protein, and that's why it's elevated in IBD patients, because it's like, yeah, you have inflammation driven not exclusively, but in part by neutrophils, and therefore you have a lot of them there, and therefore a lot of calprotectin. And then, yeah, obviously, if you don't have neutrophils there, if you have no inflammation or minimal neutrophil, then low calprotectin. That is why it's used as a marker of inflammation, like in the gut. So, it's actually produced by those white blood cells, those neutrophils. And yeah, even though it's like specific to the type of inflammation seen in IBD patients, it's not the easiest to carry out the test. As they say, it takes 1 to 2 weeks to obtain in the clinical setting, where the test is like an ELISA test. Basically, it takes some technical expertise to carry out the test. And so one of their goals is also to come up with something more convenient. So to fill this gap, we set out to engineer the probiotic Escherichia coli Nissl 1917. This is a particular strain of E. coli. I've actually personally worked with this specific strain of E. coli. And it's a probiotic. Okay. So when most people think or hear of E. coli, they think of, poisoning. That's poorly cooked ground beef. But the vast majority of E. coli are non-pathogenic and harmless. And pretty much like virtually every human being on the planet has some E. coli living, or not some, quite a bit of E. coli living in their gut. And like, it's supposed to be there, and it's largely either like doing nothing or, if anything, beneficial. And in this case, this Nissl 1917, this particular strain is actually a probiotic. It appears to actually have some therapeutic benefit when administered. So they're kind of potentially killing two birds with one stone, maybe. Where it's like, not only is this a probiotic, and they have some other beneficial effects, but they're going to engineer it to detect this biomarker, and then, yeah, maybe a dual beneficial effect of like probiotic aspect, and they're like, yeah, it now can be used potentially to monitor your disease, the severity, or your disease progression. Yeah, the clinical gold standard biomarker of gut inflammation for the goal of being used as a potential non-invasive diagnostic of gut inflammation. E. coli Nissl 1917 is a known human gut colonizing probiotic with a long track record of safety in humans and its compatibility with the canonical genetic engineering techniques for bacteria. Right. Where this known about it for a while, and it's been used for, infinite number of purposes, and it's basically amenable to genetic and the typical genetic engineering that we carry out on a routine basis, in synthetic biology, molecular biology, etc. So, we here they're summarizing basically their overall workflow. We first identified calprotectin responsive E. coli Nissl 1970 genes through RNA sequencing and coupled their respective promoters to a fluorescent reporter prior to transforming into E. coli Nissl 1917. Next, we screened those engineered E. coli Nissl 1917 sensors for sensitivity and specificity to calprotectin and optimized their performance. Finally, we demonstrated that our engineered E. coli Nissl 1917 sensor is sensitive and specific to gut inflammation in murine models of IBD and stool samples from human patients with active IBD. Our goal is to improve disease monitoring and identify at-risk patients earlier with the hope of maintaining remission and avoid irreversible bowel damage in this growing patient population. And I guess a side note, Canada has one of the highest rates, if not the highest rate, of IBD. So, going, step by step, try to explain in as much detail as possible, as reasonably possible, of like what they're doing. The broad strokes of like the genetic engineering that they're doing and carrying out, and their workflow. So, calprotectin, also known as whatever this series of letters and numbers, is a heterodimer protein produced by neutrophils, as mentioned, and monocytes that exerts antimicrobial activity through chelation of essential nutrients such as zinc and manganese, resulting in bacterial metal starvation. Right, so maybe I should start, or I can start drawing things out now, maybe. 1 second. Yeah, I guess that works. As they say, calprotectin, also known as, blah, 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 heterodimer protein produced by neutrophils and monocytes that exerts antimicrobial activity through chelation of essential nutrients such as zinc and manganese, resulting in bacterial metal starvation. So, what that means might not be the most informative diagram. I don't know. We'll see. But it's a protein, okay? So it's composed of amino acids. Not that that's super important, but it's a heterodimer. I mean, given the fact that I can draw it out. Heterodimer just means protein. Okay? They can be composed of multiple subunits. Not gonna draw out a structure of a protein because that's super complicated. But as you can have your standard globular protein, and so it's protein. But proteins can be more sophisticated, as in they can be composed of multiple building blocks, or have macromolecular complexes. And when they mean by heterodimer, so some protein, okay, are dimers, where they're pretty much have two things associating, okay? And then this entire thing is a protein, or like the fully functional active protein. And so this, if they're composed of two identical units or subunits, in this case, I can't draw two identical circles, but imagining that these are two identical circles, this would be a homodimer, as in two identical subunits come together, and this is the fully functional, protein. But now they're saying it's a heterodimer. All that to say, it's like, as the term implies, it's still two. So it's a dimer, so it's two subunits, but just two different subunits come together. And, this is just for simplicity. They may not be exactly the same size. It's just like, yeah, two different subunits come together, and both of them together as the heterodimer, that is the fully functional protein. And it's produced, by the neutrophils. And this would be how protected. And what they're saying is it exerts antimicrobial activity through chelation of essential nutrients such as zinc and manganese. That's just fancy terminology. So calprotectin serves multiple functions, okay, but it's produced by these neutrophils, okay, and those are part of the white, they're a type of white blood cell, so part of your immune system. And so that canvas. So since it's part of your immune system, it's basically involved in, your host defense system. And so one of its functions, is to sequester certain metals, okay, as they say, zinc and manganese. So all that to say, it binds to zinc, binds to manganese. I believe it also binds to iron, but they don't really highly confident that it does. And it's doing that for a reason. So it's produced by these neutrophils, and it binds and sequesters these particular metals. And this is part of your host defenses, or one of your multiple abilities of host defense. Is basically through starving bacteria from the nutrients that they require. And these are part of those nutrients. That zinc, that manganese, that iron. Just as you require iron and other metals for survival, so do bacteria, and like virtually every other microorganism on the planet. And so that is like one of the intentions of like, well, we're just going to try to starve you out, literally, by producing this protein. And then it will bind and pretty much sequester away the zinc, the manganese, and the iron, and therefore it's not available for the bacteria there. Survive and proliferate. So that's pretty much what calprotectin, let's say, the relevance here for what it's doing, and why it appears to be specific for the inflammation gut. Whether engineered bacterial sensors can be used to detect calprotectin is currently unknown, as it is unclear whether calprotectin elicits any transcriptional changes in bacterial species. Right, so this is, as mentioned, calprotectin produced by neutrophils. Well, these are your white blood cells, so it's a human protein. Okay. But what they want to do is they want to engineer bacteria that can detect the human protein. And so as they're outlining here, well, they don't even know if let's say natively any of the bacteria or the bacteria that they're going to work with even responds to the calprotectin. So it's still even an open question of like, hey, is it even feasible to get bacteria to detect this human protein? So previous work has demonstrated that bacteria species exhibited unique transcriptomes when cultured in nutrient-rich and limited conditions. Although the optimal cutoff values for fecal calprotectin that indicate active inflammation can be variable across patients, values greater than 100 grams per gram indicated the presence of inflammation with high levels of specificity without reducing sensitivity. Right, so, technically speaking, I believe as of now, there's no official cutoff of like, so yeah, it's the detection of calprotectin is not a binary. There's like a range from like, single digit numbers to like, in the thousands. And it's virtually never going to be zero. You're always going to detect something. But where is the dividing line between like, hey, you are now in like, an unhealthy state of inflammation. There's not absolutely global consensus of like, hey, if you're above this specific number, then like, yeah, something is clearly off. Of like putting it at 100 micrograms per gram. Well, if you test someone's calprotectin, it comes back at 101. Well, is that like something to actually be concerned about? Some would say yes, some no. But like generally speaking, yeah, the cutoff is around like 100 or like 50. If you test someone for calprotectin at a number of like 150, 200, it's generally thought that that is, unhealthy inflammation or that needs to be investigated. So heat maps of the statistically significant differentially expressed genes that overlapped between calprotectin treated E. coli in NISL 1917 in M9 and LB media were generated. Right. So let's see. Yeah, they performed RNA sequencing in the environments of complex and minimal media. So basically, what they did is they're working with that specific strain of E. coli, NISL 1917. Okay, and it's an open question on whether natively it can detect calprotectin. Let's say in the absence of any engineering whatsoever. Like, hey, without intervention, does the E. coli NISL 1917 detect or respond to calprotectin? So what they did is pretty much characterized its gene expression. Okay, and they characterized its gene expression under various conditions, as they say, M9 and LB. And I'm very familiar with these coli. So like this is pretty much I've actually done like exact things that they're talking about. But yeah, the difference is you can kind of think of it as M9 or sort of like starvation conditions, or like close to starvation conditions. We still want to keep the bacteria alive. And this is like, rich, sort of like eat as much as you want. Nothing is limited. Basically, the E. coli are, happy in these conditions because they're getting everything that they need and then some, as opposed to here, they're basically largely in nutrient limited conditions, and it's basically like a little bit of a stress too. They can still survive and grow, but in comparison to how much they grow and the rate of growth, in comparison to the LB, it's like, night and day. It's like they will grow very rapidly in LB, as opposed to here, it's still quick in comparison to the human timeline, but it's like, yeah, many-fold lower. But they're basically, they want to test these conditions because like Yeah, the genes that are expressed will be different in the different so principal component analysis reveals that the global gene expression profile of E. coli Nissl 1917 treated with calprotectin is distinct from untreated in both complex and minimal media conditions. Right. Let me see. Okay, yeah. And a key thing to point out is like they are growing them in the presence or absence of calprotectin. Or I think both in the presence. Double check. Treated with a clinically significant dose. Okay, yeah. So yeah, they carried out both conditions. Yeah, they had both treated and untreated. So yeah, they basically have, you can think of it as like a sort of a randomized controlled trial. Oh, they have the arm, the treatment arm, where it's like, yeah, they're growing the E. coli in both minimal and rich conditions, and then in the presence of calprotectin. And then the other arm, basically the same thing, but they're withholding calprotectin. They basically want to see the differences in gene expression that occur when calprotectin is present or absent. Because if there are changes in expression when you're comparing whether the calprotectin is there versus not there, then that strongly suggests that the bacteria are responding in some way to calprotectin. And then that strongly suggests that, they are sensing it already in some way. So overall, there were 2,200 differentially regulated transcripts from calprotectin-treated E. coli Nissl 1970 in M9 media compared to 1,200 in LB media. Way more gene expression when the calprotectin is there, and then when the E. coli are grown in those close to starvation conditions. And then, yeah, in comparison to basically about half or 1,000 less when grown in those, rich, all-you-can-eat conditions. But there's some overlap with about 800 overlapping genes. And these results show that calprotectin elicits a robust transcriptional response in E. coli Nissl 1917 that could be used for the construction of the synthetic sensor gene circuit in our probiotic-based whole cell biosensor. So yeah, as there's pretty much, this is like a proof of concept, and basically, yes, this appears to work, or has potential to work, in that it appears like the bacteria, when we tested them, they are responding the presence of the calprotectin. When we grow them in the presence, whether in the rich conditions or like the limited conditions. So, functional classification of differentially regulated genes by gene ontology analysis indicated enrichment of genes in pathways involved in ion transport, cellular metabolic processes, and cell motility. Interestingly, the upregulated genes with the highest fold change were involved in cellular regulation of zinc ion. This includes the expression of genes, well, these both under control the same law promoter, yes. Well, basically they narrowed it down to, a handful of genes that it appears like, they were responding or generating some sort of response to the calprotectin. And as they explicitly say, we identified 5 genes with both the highest fold change and statistical significance after calprotectin treatment that were consistent between the two groups. The promoters of these genes were then screened as potential targets to be used for construction of the biosensor of calprotectin. So, they narrowed it down to 5, okay? But they don't just want a gene that, responds a little bit to the calprotectin. Again, they're using this for reporting and tracking, okay? So that's why they're also looking at not only as a binary of like what is and what is not expressed but what is the level of expression? They basically want something that, like, when it turns on a lot. Basically, they want a bright light, and sort of coincidentally, they're going to use light basically as one of their reporters, or like as, well, yeah, the reporter. And so it's like, yeah, they don't want to use a gene that results in a dim light. They want to use a gene that results in a bright light. But they don't have to look, they don't have to zoom in super close saying, hey, our biosensor is working. They want something obvious. They want something that's going to generate a lot of the reporter that they want to use. They engineer it. That's why they talk about, basically differences in expression. The expression of G. Because it matters the level of expression. They want something with, like, high expression. And that's why, from those 5 genes that they narrowed down, they did a little bit more narrowing down, saying, hey, of these 5, which one appears to be, or which 2 of these, I think that they narrowed down, have the highest expression. So, to identify potential biosensors for calprotectin, the respective promoters of the 5 most significantly upregulated genes were coupled to super folder green fluorescent reporter protein, or SFGFP, prior to transformation into E. coli Nissl 1917. These recombinant strains of E. coli Nissl 1917 were then treated with 100 grams per gram calprotectin to assess for the sensitivity of these promoters to calprotectin using SFGFP fluorescence intensity as a surrogate marker, figure 2A. So I'm going to take a closer look with that. Yeah. Let's see. Oh, this is not figure 2A, that's figure 1A. Figure 2A is right here, yeah. If I can get up one moment. This second. Going 1 second. Okay, yeah. Yeah, so we're looking at basically the figure here. What they're doing. So with their, 5 identified, okay, they basically do some engineering, and they basically include this SFGFP. So this is like their promoter GFP, composed of those 5 genes that they identified. they did that initial experiment of like growing the E. coli in the presence of calprotectin, and then in those different presence or absence of calprotectin, and then in the different conditions of both rich and minimal conditions. And then once they narrowed it down, now they want to see like, okay, can we actually, or rather, which one's the strongest, and then which one can actually act as pretty much like our actual reporter that we're gonna go with. They haven't narrowed it down to like one single thing. So to do that, to see like basically which one, shines the brightest and gives the strongest signal, and that's why they couple it to this GFP, this green fluorescent protein, but it's a variant of green fluorescent protein, presumably probably a better variant. It's SFGFP. They call it superfolder, which I'm not familiar with. I just kind of assume that is probably just like whatever, a better version of GFP. Not that it matters all that much. What matters is like, yeah, they're using a fluorescent protein, and they're going to use pretty much like the levels of fluorescence, and like basically to assess which one of the 5 that they narrowed down, which best one. So like the more fluorescence, the better it is. The more green fluorescence, the better it is. And so that not a lot of space to write. Learning curve, tablet. I guess I'll leave it small. So yeah, what they do, sort of as depicted here, okay. So each gene will have, so that this little black box here, okay, that's gonna be like, not black, but that's like that promoter. And that's why it has that little red arrow top of it, basically saying like, hey, sort of like the gene starts here. But this basically acts as a regulator. Now, when it comes to gene and gene expression, we don't want things on all the time. We want to be able to turn them off. We want to be able to control the levels. And so like, this is one of the ways of like regulating. So they're basically looking at or assessing like which one is like the strongest promoter, which one basically gives sort of like the best on signal. Obviously my right, not the best on the tablet. I'm gonna get used to that. So yeah, they have 5 of them, okay? And how they're going to assess their on signal is like through that FP. That's essentially what they do here. So they have here, those are their genetic constructs, okay? These are what we call circles. These are called plasmids. And these are basically circular pieces of DNA that, we can pretty much easily get them into bacteria. That's essentially what they're doing here. As you can hopefully see, it's like each of these plasmids will be, the terminology, be transformed into the bacteria, and then they will either add, they will either add or leave out the calprotectin, and then they are going to assess basically the levels of GFP. Basically, the more the better, because like this promoter sort of acts as their on signal, and they want the greatest on signal. And you can see that there are differences. And so in red, that's in the presence of calprotectin. So what matters most, or I guess maybe not most, but what matters a little bit more is like the red lines. Okay, and you can see that there are differences in the red lines. So even though all of the 5, okay, they all respond to calprotectin, they basically result in, green fluorescing from the GFP. But there's clearly one that stands out, where over time, the x-axis, you have, basically the top one, maybe use a different color. One of them stands out as essentially a winner, where it produces the most on signal. That's basically what they're interested in, and why they're carrying out this experiment. So yeah, that's pretty much like the workflow of what they carried out, at least for this experiment. So where was I? Yeah. So these recombinant strains of E. coli NSO 1917 were then treated with 100 micrograms per gram of calprotectin to assess for the sensitivity of these promoters to calprotectin itself using SFGAP, fluorescence. Yeah. So we initially screened promoters of the top up-regulated calprotectin responsive genes, like these 5 here, which are all induced in the setting of zinc starvation. Interestingly, the expression of these genes is regulated by transcription factors from the ferric uptake regulator family that are responsible for cellular homeostasis of metal ions, including manganese, nickel, zinc, and iron. So even though these promoters, appear to be responding to the calprotectin, they're pretty much saying sort of like what their native function is. Of like, hey, these respond to metal ions. Because again, as an example, and as I alluded to earlier, just as we require metal ions like iron, for survival, for growth and survival, so do microorganisms and bacteria. And so, very often, rather virtually all the time, bacteria, they will have genes that are responsible for basically acquiring, basically eating, so to speak, metal ions from the environment. Although they obviously don't eat the same way that we do. But they take in and consume metal ions from the environment. And so they have genes that are devoted to that, as they say. Cellular homeostasis of metal ions, such as these ones, and they include iron here. So like those genes that are responsible for, allowing them to get some of these ions, for example, iron, they want to be on all the time, okay? So just as, even though we as humans, we need iron to survive, we don't need to be consuming it 24/7. We need regular doses. The same principle holds true for, virtually all microorganisms. Of like, they don't want to be consuming, iron from the environment all the time. They want to, consume it when needed. And so the genes that allow them to uptake it from the environment, they need to be controlled. They don't want to just like make things for no reason. And that's why they will be responsive to their environment. I guess I should actually, again, draw it out. Sort of a good example, arguably, if I have any area of expertise, this one of them, is pretty much how do E. coli take up iron from the environment. So, using iron as an example. That is like what I'm most with. So, you know, the E. coli will have a gene, likely multiple genes, devoted to, iron uptake. And what do we mean by iron uptake? Pretty much like it'll allow them to make a certain molecule, and that molecule will bind to iron. But you don't want it on all the time. So, it'll be away regulated. And they even explicitly say it, and that's why I highlighted this ferric uptake regulator. So these genes, will be regulated. That's why you have a ferric uptake regulator, where these genes responsible for iron uptake, they will be under the control of that ferric uptake regulator. And how it's controlled, it's like, when there's a lot, of iron in bacterial cell, iron, this, off. And thus, like, pretty much turning that off, because, like, since there's iron there, you don't need to be making the genes that allow you to get in more iron. And that is controlled through this ferric uptake regulator. They actually call it like a fur box, where it's like in the presence of iron, there is, something that basically blocks expression of the genes that allow it to uptake iron from the environment. But if it gets to the point, using the example in the paper of like they've grown the iron in the minimal conditions, it'll be starved for iron. The iron will go away, and then pretty much that will turn this. Now, on condition. It'll basically, it'll remove the blocking of the gene expression. It will allow it to make these genes for iron uptake. So, and the same principle holds true likely for all the other metal ions. You don't want, stuff on all the time. So, the promoters of these genes are all under the control of the transcriptional repressor zinc uptake regulator, or ZR. So this ferric uptake regulator, that's FER. And so for this other one, you have the zinc ZR. While the promoter of TON-B is regulated by the transcription factor ferric uptake regulator, right? Or FER, like I mentioned. These results are consistent with an expected mode of action for calprotectin sensing involving zinc chelation, leading to metal starvation. We first evaluated the responses of biosensors constructed from these promoters in minimal media, or M9, with the goal of identifying promoters with high sensitivity to calprotectin and low background induction during the growth phase. Right, so they want, in addition to, something that's obvious and that will be easily characterizable. Second, they also want something that is basically a binary. But like, hey, when it's off, it's off. Because again, they're using, in this case, starting out, they're using green fluorescent protein. And so if that's going to be their reporter, they don't want false positives, as in they only want to get a signal when there's, calprotectin present. Uh, and that's why trying to basically ensure or talking about like this background or low background induction of like, hey, basically we don't want uh, these genes on and making GFP when there's no calprotectin present. That would lead to basically a false positive of like, hey, we're getting green fluorescent protein, but there's no calprotectin present. Because again, the ultimate goal is like this is sensitive to the detection calprotectin biomarker. So that's why they want to identify promoters with high sensitivity to calprotectin and low background induction during the growth phase. Basically, saying, like when it's off, it is truly off, and then when it's on, it is on. On signal, it's only coming from the presence of calprotectin. E. coli Nissl 1917 transformed with plasmid containing promoters, and these like I said, those circular pieces of DNA, those are the plasmids. Basically, the engineered genetic material that contains, that promoter, the different promoters with that GFP. And ZIN-T demonstrated calprotectin-dependent activation of SF-GFP fluorescence compared to untreated controls with similar growth patterns. That's figure 2b right here, but more or less roughly the same thing that I covered. But it's just now we're going, gene by gene. And yeah, as you can see, there are differences. So in red, that's the presence of calprotectin. So yeah, it seems like this ZIN-T ECN, pretty much the third from the left, I think it appears to basically generate the signal that they're looking for. Yeah, where if you can see the other ones, either in the presence or absence of calprotectin, here on the far left, that still seems to generate a GFP signal. And then, like, yeah, for this one, better, but you still get some signal, the absence of calprotectin. As opposed to here, pretty much, you only get a signal when you really have presence calprotectin. So yeah, ZIN-T ECN displayed. Oh, they just go through all what I just went through of like the differences in the fluorescence of like, yeah, you get high signal here, but still get some signal in absence of calprotectin. As opposed to like, yeah, in ZIN-T1 might not get as super high of a signal, but the advantage is like, yeah, it seems like it's mostly a binary. When it's on, and when it's off, it's off. And that's why, yeah, thus, only YKG-MO ECN and ZIN-T ECN remained as viable candidates for our sensor. Yeah, no surprise. They further evaluated in the middle, basically, which makes sense. Because the other ones, yeah, it doesn't seem like they're not super sensitive. Calprotectin. You'll get a signal when the calprotectin is left out. So next we evaluated whether calprotectin sensitivity in YKG and ZIN-T was also observed in complex media conditions. So yeah, they started off with minimal, close to starvation conditions, and now they're going to see how it stacks up rich, sort of all-you-can-eat conditions. So when treated with calprotectin in LB, ZIN-T ECN no longer exhibited its robust calprotectin-dependent activation of SFGP, figure 2D. Okay. Interesting. Okay. Yeah. Show it. I have it. Second. No. Have it. Oh, that didn't matter. But basically, it's just here, figure D they're referring to. And they're like, yeah. In the black checkered line, here's where they're growing it in like the rich conditions, the all-you-can-eat conditions. And basically, what they're seeing, what they saw up here with the Zinti, it's basically now behaving differently. Of like it now, it's no longer as responsive to the calprotectin, or like you're getting essentially a false positive when it's grown in the rich conditions. Where? Yeah. While the YKG maintained its response with a 23-fold increase SFGAP fluorescence compared to untreated control, or 2E. So yeah, this one here, looking at the YKG MO. So fold change over untreated. So even though in the minimal conditions, the YKG, even in the absence of calprotectin, it was generating a signal. But in presence of calprotectin, the signal was much higher, and it's like when it was grown in the rich conditions, it just like further amplified the signal, or the discrepancy in signal, whether in the presence or absence biomarker calprotectin. So growth conditions were similar for all groups in this study. Interestingly, untreated YKG did not exhibit a similar increase in SFGAP production compared to when grown in minimal media after 13.8 hours. This effect is likely due to lack of metal ion starvation. Yeah, important. So the intergenic region upstream of genes can contain a variety of functional and regulatory elements, including binding sites for transcription factors. In addition, the zinc-specific regulator ZR has been shown to bind to sites, the intergenic region upstream of known promoter sites of other zinc-responsive ribosomal proteins. Yes. So to improve the stability and reduce background expression levels of the YKGMO promoter, the entire intergenic region upstream of the YKGMO gene was identified and coupled with SFG new strain of calprotectins. Okay, yeah. So they basically carried out, further genetic engineering / manipulation, because even though this YKGMO appears to be overall the best performing in both conditions, both in the starvation conditions and then in the all-you-can-eat conditions. Although again, not truly starving conditions, they're still able to grow and thrive. But they basically, as highlighted here, you still get, carrying the solid red lines, you still get, the solid red and the solid black. Still get some baseline level of expression, some green fluorescing when the calprotectin is left out. So they basically did some optimization, further optimization to reduce that background signal. Because again, they actually want a binary, and they don't want not a binary, because they do correlate it with the actual levels and give it a numerical value. Basically, they want to minimize, if not ideally eliminate, false positives of like when the calprotectin is absent, it is actually off, and like as close to fully off as possible. The expression of the GFP is off. And that's why they pretty much did further genetic engineering. And as they say, and as I highlighted, this new strain exhibited a 68% reduction in background SF-GFP expression level when untreated in minimal media, while maintaining similar induction when treated with calprotectin. Sensitivity to calprotectin in complex media for YKG was comparable to calprotectin-treated YKG. So the other one. Growth conditions were similar for all groups in these experiments. Changes in fluorescence can be detected as early as 1 hour post-treatment of calprotectin in YKG compared to untreated. So it seems like, it's very fast, as in like, once the bacteria there are growing and they add the calprotectin, seems like, they're turning on the genes and getting green fluorescing within like an hour. We also performed a dose response curve using the YKG, the now new improved YKG strain with varying doses of calprotectin. Right, because again, they don't want to just be able to say whether calprotectin is like there, present or absent. They want to be able to say like how much is there. Is it a lot? Is it a little? We found that the lower limit of detection was 25 micrograms per grams of calprotectin, with maximum SFGP signal increased with higher doses of calprotectin. Yeah, there's this dose-response relationship. The more calprotectin that they add, the more green fluorescing signal they get. Taken together, our results show that this new improved YKG is a viable candidate for in vivo sensing of calprotectin. So, as of now, they are working in vitro. Okay, they start off with sort of a proof of concept, trying to narrow down genes, candidate genes, and then they characterize those candidate genes, and narrow it down like, hey, what are the set of genes good candidates, and from there they further narrowed it down to like this one specific gene that they optimize. Now, new and improved YKG. Calprotectin sensing E. coli can reliably detect gut mucosal inflammation in vivo. Now they are moving up to in vivo. So to test whether our calprotectin sensor detects gut inflammation in vivo, the YKG promoter was coupled with Lux cassette, not going to say our alphabet, and transformed to ECN to generate a new inducible bioluminescent calprotectin. ECN. So they're overall doing the same thing, but they're switching it up a little bit. Okay. Now, instead of using this GFP, presumably improved superfolding GFP, they are moving to a Lux, bioluminescent. GFP, fluorescence, or they were using fluorescence, and that's a specific phenomenon that, I'm not going to give a lesson on fluorescence now. Well, all that to say, they are now actually changing to going from fluorescence to bioluminescence. Okay, and the difference here overall is, bioluminescence is the actual generation of light. Okay, even though it's GFP, okay, and there's fluorescence, and you can kind of like basically see some green fluorescing. You get bacteria to make GFP, but it's not quite the same. You're not actually generating light, you're fluorescing, which is a different phenomenon than light generation. Where here it is actually like light that you are seeing. Actual sort of, not true flashes of light, but like actual light generation, as opposed to fluorescence, slightly different, if not loosely different, scientific. So, because basically, for fluorescence, okay, you have P, on the y-axis here, they're measuring something like specific. Fluorescence, again, is a specific phenomenon, without getting into the nitty-gritty details, at least not right now. And so, yeah, they want to move to something that's even more convenient, basically. And just the generation of simple light through bioluminescence is just way more easy and convenient. And that's why they are switching up the system. Where fluorescence, totally fine for in vitro, okay? And, they can just, take a cell culture of E. coli and then see how much it fluoresces. As opposed to, in this case, now that they're moving in vivo, and I believe in vivo in mice, a little bit more challenging fluorescence. And so they're moving to something a little bit more convenient, if not a lot more convenient, in bioluminescence and just the straight generation of light. Just go by light intensity. Or an easier way to measure. So, this strain of ECN is capable of endogenous production of bacterial luciferin and luciferase to generate a luminescent signal on whole animal imaging when the YKG promoter is induced. So these are the ingredients for this bioluminescent phenomenon. You have luciferase, okay, and that's basically a class of enzymes, if I remember correctly, which I believe that I do. I don't. Yeah. So, class of enzymes, and then the luciferin, like basically what the luciferase uses to make the light. They're to generate light. Yeah. The terminating for pneumology. Yeah. So, yeah, this luciferin and luciferase, basically when they come together, the luciferase acts on the luciferin, generate a light. Or I guess to use more technical language, a luminescent signal. And then now they're using whole animals, so whole mice. So CS ECN Lux demonstrated robust calprotectin-dependent activation of luminescence compared to untreated controls with similar growth patterns. The maximum luminescence of calprotectin-treated ECN Lux was about 1800-fold over untreated controls. So basically, all that to say, it's like the system is still working. Pretty much what they saw with the GFP, they're now seeing with this bioluminescence. And the lower limit of detection of the Lux, the ECN Lux, is the same as previous experiments with, pretty much they can get down to 25 micrograms per gram of calprotectin. Like, that's way in the healthy range. Basically, as close to, like, healthy levels of calprotectin, indicating virtually no inflammation. So, to model IBD in mice, we utilize the dextran sulfate solution, DSS-induced colitis model. So, yeah, pretty common in IBD research, or rather using mice for IBD research. They just, they basically get them to consume, basically, not so nice stuff that will just make their GI tract in. Because, that's the hallmark of IBD. Inflammation in the GI tract. Not only the GI tract, that is generally the hallmark, uh, whatever, physiology. So, and they induce it by, giving an oral gavage of 3% DSS daily for 7 days. Basically, kind of don't treat these very well for 7 days. Bunch, sort of force feed a bunch of DSS. And we chose the DSS-induced colitis model mainly due to its ability to rapidly induce colitis phenotype, and also extensive evidence demonstrating that DSS treatment resulted in elevation of fecal calprotectin levels. Starting on day 5, we orally gavage the control and DSS-treated groups with either the Lux or the ECN without sensor daily. Yeah, they basically started, force-feeding their engineered bacteria to some of these mice that had, colitis induced in them forcibly. But then obviously as a control, okay, they just gave run-of-the-mill, unmodified E. coli that will not generate light or the bioluminescence. That is their control group. So, in vivo luminescence imaging was performed using the in vivo imaging system on day 7. Luminescent signals were detected in DSS-treated mice that were gavaged with the Lux, with signals reaching, 1 × 10⁵ radians. They're actually measuring the amount of light with the in vivo imaging system. That was significantly increased compared to those measured from mice not treated with DSS and not gavaged with the Lux system. And this is, figure 3B. Yeah, okay. So, probably think I have that particular figure. 1 second. Yeah. So I mean, pretty much, this is what arguably matters, like, the most. So, they have their multiple different mice treated in different ways. This mouse here, basically, they don't have the DSS, they don't have the lock system, so it's basically a negative control. Okay, so there should be no inflammation. Okay, but even if there was inflammation, you're not going to generate a signal because the lux thing that generates the light is absent. And then pretty much the second one is also overall a negative control, because you're not going to get any light generation because there's no DSS, so there's no inflammation. So that's like promising in the sense that, it has the lock system, but it's remaining off because there should be no inflammation. And if there's no inflammation, there should be absence, or the calprotectin should not be present. Therefore, you don't get the luminescent signal. And then pretty much, the third mouse here, basically, just the flip side of that, where it's like, there's inflammation, but you're not going to get any luminescence because it doesn't have that lux system, engineered E. coli that generates the bioluminescence. And then, as highlighted the last one, the fourth one, you have both present. They have both the engineered E. coli, and they've forcibly induced inflammation in the mouse. And then, you are getting the bioluminescence. And then now they're just like assigning numbers, actual radiance numbers to it. it's the fourth one that matters the most, where you get the most radiance in comparison to like all the other three. They're basically negative control. Ideally you shouldn't get any radiance, but no system works perfectly, and you always get some sort of baseline. But yeah, the other ones are just like way higher. So pretty much their system is working as intended in vivo, in mice. Importantly, this SFGFP positive population was not seen in the absence of DSS or in mice that were not given the CSCSP. Second. Oh. Yeah, I missed. Oh yeah, to further demonstrate that our calprotectin sensor is capable of functioning in the complex environment of the mammalian gut. I got... Oh yeah. I got around here. But it's pretty much summarizing what I already went through. Of like, yeah, luminescent signals were detected in DSS-treated mice that had that Lux system. And so the mouse colon collected after 7 days of DSS treatment had altered colonic architecture, goblet cell loss, immune cell infiltration compared to controls, confirming the presence of gut inflammation. So yeah, after running the experiments and after, looking how much light or luminescence was generated, okay, they want to actually confirm, or actually further confirm, the system is working. In that, based off of the signal that they're getting, okay, it's saying that, hey, inflammation is present in this mouse. Okay. The one that, had the DSS basically force-fed to them, and then had the Lux system also essentially force-fed to them. And then they got a signal. So that should indicate that there's inflammation, a lot of inflammation occurring in the GI tract of this mouse. But they're not just going on faith. Actually, went in and like basically dissected, cut open the mouse. Okay, let's look at the GI tract. So our system and the signal is saying like there's inflammation there. Let's go and basically visually confirm it. That's what they did, which is important. And that's why that they explicitly say confirming the presence of gut inflammation. So it's like, yeah, the system appears to be working so far exactly as intended. Like the system that they designed said that there's inflammation here, and a lot of inflammation, and then when they go and look, there is actually inflammation. And serum and stool calprotectin levels were significantly elevated in DSS-treated mice as well. Figures S3. Oh, yeah, that's supplemental. There. So further confirming that luminescent signals are corresponding elevated calprotectin and gut inflammation caused by DSS treatment. To further demonstrate that our calprotectin sensor is capable of functioning in the complex environment of the mammalian gut, utilized the previously created YKG, now going back to GFP reporter strain, transformed to ECN, termed, yeah, further in vivo studies. Okay, so now it looks like they're going back to the GFP system. And then now, yeah, they pretty much carry out the overall same process. But now they need to use a slightly different measurement. Since they're going back to fluorescence, okay, as opposed to luminescence, they have to use a different instrument to measure it. Because fluorescence, not just visual. Especially when it comes to looking in. But they basically use flow cytometry to basically analyze the stool samples of mice treated with the DSS and the engineered GFP, and basically they're getting more or less replicating the same results. Like they're getting green fluorescence when when there's inflammation present and when the mouse has the engineered E. coli with the GM. Basically, looking at this graph E here, basically on the far right, where it's like it has both the engineered E. coli with the GFP. Now they're looking at green fluorescent. So they're just confirming that both systems work, but they have to use different tools to measure for bioluminescence, flash radiance, and then here going back to fluorescence or GFP. And they're using flow cytometry without getting into the nitty-gritty details of cytometry. Basically, fancy counting of cells, okay? And they're just going to count the cells that are fluorescing. In this case, the E. coli cells. That would be contained in the stool samples of the mice. So, importantly, this SFGFP positive population was not seen in the absence of DSS or in mice. Yeah, exactly. So pretty much, their signal is specific. They're only getting fluorescence when inflammation is there and when, the GFP with from the engineered E. coli there. So furthermore, DSS treated mice that were fed, the E. coli GFP had 26-fold increase in the SFG that were also fed. Yes. So contrasting. Yeah. This demonstrates that E. coli GFP is highly specific for calprotectin, even in a highly heterogeneous environment such as the mouse colon. Thus, both sensor strains are activated in response to gut inflammation in the mouse colon, and this signal can be detected in samples. So, pretty much, no matter which way that they do it, either with the GFP or with the bioluminescence, both appear to be viable when moving up to in vivo in mice. Pretty much adding further confidence to the system. So, now I believe they're, sort of highlighting, they're going to scale up now further in vivo, because again, the ultimate goal is like, does this work in patients? Oh, is this actually a viable strategy to use, in a human patient setting with individuals that have IBD or diagnosed with some form of IBD? So, calprotectin sensing E. coli can differentiate stool samples from patients with active IBD and those in remission. So to evaluate whether our engineered E. coli GFP can identify patients with active IBD, we collected fecal samples from patients undergoing stool studies, either at the emergency department, in the hospital, and in outpatient clinic settings. Stool specimens were collected from 17 patients that consisted of 11 patients with a history of IBD and 6 with no history of IBD. Of those 11 patients with IBD, 4 had a history of Crohn's disease, 7 had a history of ulcerative colitis. Fecal calprotectin levels were ordered for these patients for the indication of disease surveillance, therapeutic drug monitoring, or a suspicion of acute IBD flare. Right, so they're pretty much saying, well, there are some patients that didn't have IBD, I believe. No history. 6 had no history of IBD. Then 11, they were basically, being assessed, or they were basically undergoing the test for calprotectin. Okay, that's used, in the hospital setting as part of management of their disease or tracking of their disease. And they're pretty much like saying, highlighting some of the reasons why this biomarker is measured. Patient population or disease surveillance, like, hey, is your disease active or not? Therapeutic drug monitoring, which is, they like, is the drug or the therapy actually working? Socrates, welcome. Thank you so much for the raid. Super kind of you. And great to see you again. And also, shout out to you. And also, Deflux, welcome. Long time no see. It's going well. And iCal, welcome. Great to see you as well. But yeah, so I hope you're doing fantastic, Deflux stream. Oh, well, I guess now you are a streamer, which I didn't know that you're now a streamer. I should probably give you, rather, I will give you a follow. And I don't know. I might be a therapeutic drug in inducing sleep. Maybe. Oh, software dev. Okay, super cool. You know, I'll probably check it out, even though I have zero experience with software dev. But I'm hoping the streaming and everything else is going well. Great to see. Yeah, I think you're like one of my first followers, going back over a year. But right now we're going over this paper here in detail. Basically using engineered bacteria for basically patient monitoring, or with the goal of patient monitoring. And yeah, Socrates, hope your stream went well. Thank you again for the raid. Super kind and super generous. So, we're going right now. We're going to the section. Basically, they started off in vitro, and then they went into in vivo, but with mice, and now they're going up to humans. Like, does our engineered bacteria, okay, that we want to use to monitor a biomarker for these patients, does it actually work for these patients? So, for IBD medications, patients in the active IBD and IBD in remission groups were on a mix of therapy ranging from, well, yeah, not super important. Of note, many of the patients also were taking oral multivitamin at the time of stool sample collection, which includes, yeah, zinc supplementation, blah, 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 yes, yes, yes. Okay. So, of the patients with no IBD history, fecal calprotectin levels were evaluated for indications of diarrhea, rectal bleeding, or abdominal pain, with laboratory results of calprotectin levels of less than 50 micrograms per gram for all specimens. Oh, Admiral Gims, welcome. Good to see you again also. Hope you're doing well. So, yeah, we covered the artificial... did I? It's totally possible. I spelled artificial wrong. Yeah. Totally spelled artificial. No, actually I didn't. I could. Now I'm like, second guessing myself. Yes, I totally spelled it wrong. Thank you for pointing that out. Well, I like to think I can spell, but obviously not. That's super appreciated. Oops. But yeah, we went over the paper with these artificial embryos. We didn't go, well, we went over the article and briefly touched upon the paper. It's this paper here. Complete human day 14 post-implantation embryo. uh, which, as I went through, I think introduces a lot of ethical questions or ethical problems for quite a few people. Or as I went through, some people put the dividing line of personhood of like implantation in the uterus. But what happens when we get to the place of like we can make an embryo requiring no implantation? And then also I covered some of the research about artificial wombs. So again, might be getting to the place like 30, 40, or maybe 20 years of like, artificial embryos combined with artificial wombs. Crazy to think about. And the ethical problems that I think it might pose for quite a few people. And thank you for the kind words. I appreciate it. Kind. Yes. So they're looking at both patients that should have inflammation in their gut as part of their disease. And then they're also looking at patients with no disease. Okay. So they have a mix. They have people, okay? People, including patients that have high calprotectin, and then people that have low calprotectin. So it's not a lot. I think it's like about 20 or so in total. But basically, enough for a good proof of concept. And they just go through some of, the simple demographics of some of the people or the actual patients. Where you have some in remission, okay, and that's corroborated by their low calprotectin levels. And then you have ones with actual active inflammation, hence active IBD. That's corroborated by their high calprotectin levels. Now they're going to use their system on this group of people. Because it should be representative for exactly what they want. Of like, does it pretty much tell us like, hey, this person doesn't actually have inflammation? And does this patient that's, in and with active IBD, do they have inflammation? Can we detect it? And how much can we detect it? Can we actually put a number to it? To determine whether their engineered E. coli with GFP is activated in response to stool with elevated levels of calprotectin, we co-cultured their engineered E. coli GFP with stool samples from patients with no IBD, IBD in remission, and active IBDs. Figure 4A. Yeah. So this is not fully in vivo, at least not in my opinion. So before, when they were using mice, okay, the mouse model of IBD and inflammation, they actually put their engineered bacteria inside the mice. They basically force-fed it to them, and then they induced inflammation in these mice. Okay, so like as highlighted in the figure, they're looking at the mice and they're getting a signal inside the mice of like, hey, this mouse is lighting up, showing that our system works in the mice. But here, they're not actually giving their engineered bacteria to the people, okay? They're just taking the stool samples from the person, and they're just like, putting it in together, in like a flask or, some sort of controlled environment with their engineered bacteria. So it's a little bit different where the mice were actually force-fed the engineered bacteria. This is not the case, either voluntarily. People aren't consuming the engineered bacteria. A little bit different. So it's not fully in vivo, just as they say, co-culture. So immediately after co-culture, the GFP fluorescence normalized to optical density at 600 nanometers. I highlighted this because it is like 99% certain a mistake. It would be nanometers, not millimeters. Was comparable for the samples from patients with no IBD, IBD enrichment, and active IBD, right? So, they co-cultured and, they pretty much, with their flasks filled with they're engineered bacteria with the green fluorescent protein. So they dump in the processed stool samples, and then things just like uh, basically uh, settle to baseline. And then from there, after 12 hours of co-culturing, okay, the E. coli GFP co-cultured with fecal samples from patients with active IBD, so the inflammation should be there, had significantly increased OD 600 ratios compared to those that were cultured with samples from patients without IBD or IBD in remission. So basically, the ones that should not have or actually don't have inflammation in their gut. Pretty much saying that their system is working. The figure 4b, which is, yeah, here, what matters most is basically the last column after 12 hours is active IBD. So basically saying these individuals here, and they should have, the high levels of the biomarker, the calprotectin, indicating inflammation in these people. And that's essentially what it is saying. After 12 hours, they see a signal of their green fluorescent protein when they co-culture it with engineered bacteria. And then when there's no disease, so no inflammation, and then when the IBD is in remission, so the disease is under control, and you have basically very little, ideally virtually no inflammation, or their inflammation resembles no disease. That's reflected by the numbers a little bit. Okay? Ones with no disease, you get less than 5, or like, basically single digits. Maybe at most mid double digits. As opposed to those with, IBD in remission, low, very much lower than thousands or hundreds. But it's not quite reflecting those with no disease, no inflammatory disease whatsoever. But still, pretty close enough. And their system basically says, is indicating the same thing. Like they're only detecting inflammation with those with active IBD. They have those high levels of the calprotectin biomarker. So, there was no significant difference between the GFP ratios when comparing patients with, already read that. So flow cytometry of their engineered E. coli GFP co-culture with fecal samples from patients with active disease confirmed the presence of a distinct GFP positive population of bacteria as early as 1 hour of co-culturing and saturation of GFP positive patients. Yes, not super. In addition, there was a faster rate of activation of GFP positive populations for stool samples from patients with active IBD that had higher laboratory measured calprotectin. So, overall, this validates that their engineered E. coli GFP can effectively sense elevated calprotectin in human stool samples, and it has the potential to be used as a reliable and efficient method of disease activity surveillance in IBD patients. So basically, almost, not quite, but almost full proof of concept. Although, I don't know if their actual end goal, like having people, well, have the bacteria inside their gut, living there natively. Because, like, again, with the mice, basically force-fed their engineered bacteria, and it was residing there in the gut. So maybe that isn't their end goal. On something, they can take the stool samples of the patients, and then just, like, process it in a lab. Which I guess that would make more sense. Because, yeah, so I guess it is, like, full proof of concept. We have developed an engineered probiotic that, and yeah, again, going back to the strain of bacteria, E. coli, that they're using, it's this E. coli Nissel 1917, and that, as mentioned multiple times, it's a probiotic, and it has sort of been trialed in the treatment of IBD. And, like, yeah. And, well, it's called E. coli initial 1917. Surprise, because that's the that it was basically discovered. But it's been used as a probiotic for quite a while. So it might have potentially this dual effect, maybe if they actually want to administer it to people. Where it's like, yeah, we can now use it to monitor your disease, and maybe you might have some benefits from consuming the probiotic. Although, from what I remember of the literature, the research, yeah, the results aren't dramatic, or it's not like this, super significant probiotic. Yeah, it might be beneficial. So, yeah, we have developed an engineered probiotic that can reliably detect calprotectin, clinical gold standard biomarker of non-invasive gut inflammation, with sensitivity and specificity for the purpose of disease activity monitoring in IBD. Oral delivery of their engineered E. coli GFP resulted in the presence of a distinct GFP and bacterial population in stool of mice with DSS-induced colitis. So again, mouse model of colitis. So basically giving them a bunch of toxic stuff that generates inflammation in their GI tract. These GFP bacteria were not present in mice that were either not treated. Oh wow, Struggle Session Radio, thank you so much for the raid. Welcome raiders, first of all, as well. That's super kind of you. And also shout out to you. That might be cooldown. Okay, yeah. And welcome Viceroy Xerxes. And also give you a follow, Struggle Session Radio. I hope you had a great stream. And yeah, welcome. I am Corey Campbell. I'm a biochemist. I cover science, research, and science news. Right now we're going over this paper, basically using engineered bacteria to basically monitor these IBD patients. And we're pretty much just going over the summary of their results in the discussion, where it's like, yeah, they engineered this E. coli that produces basically light or fluorescence, and it basically allows them to monitor this particular biomarker in these patients, where they can actually, see if the biomarker is increasing or decreasing. And, it correlates with basically how active their disease is. And yeah, they started off in vitro, and then they worked up to mice, and then they actually worked up to using it in actual humans. And it appears to work quite effectively. So, but yeah, can't thank you enough for the raid. Super generous and kind. And yeah, I really hope you had a great stream. And yeah, I definitely will check out your stream. Doom, you also stream science and tech? Check quickly. Oh, got it. Non-smoker. Okay. I know exactly who you are. Yes. Basically a politics streamer, which I definitely cover some of that as well. And welcome, Schlatsky. And I definitely seen you, I believe, on Wick's channel doing some debates and on some panels. One second. But yeah, hope you had a great and well. Also appreciate, I guess, yeah, name change or the brand change. But welcome. Thanks again for the raid. And thank you so much for the follow. And Twin Tower Enjoyer, also thank you for the follow. Super appreciate it. Happy to have you here. So wrapping up this paper. Yeah, consistent with our
flavor country.
Oh no. I am a very radical lefty, and you have nothing to apologize for
Welcome to
at
Flavor
all.
Country.
Like I've checked out, I've seen quite a few of your debates and discussions. I believe mostly on Wick's channel. Like I'm way super left comparison to Wick. So yeah, super kind for the raid, and I think there's a lot of overlap in our politics. Maybe not 100%, but I think pretty darn close, from what I understand and from what I remember of some of your debates and discussions. So yeah, wrapping up this paper, and thank you for the follow, Struggle Session Radio, super appreciated. So our data from clinical stool samples showed that sensor activation was only present when cold cultured was stool from patients with active IBD. Importantly, SFGFP of our sensor can quantitatively track with the laboratory measured levels of fecal calprotectin. What this means is basically, you have your traditional way of measuring this biomarker, okay, this calprotectin, which is a little bit more convoluted and time consuming. So not only does their system work, okay, and they've established proof of concept, but they've also pretty much demonstrated that it is equivalent to gold standard that is currently used, but basically just like even better, where they're able to reproduce these numbers, but basically reproduce them on a faster time scale. Because as they're going to cover when wrapping up, the traditional way to carry out the test, measure this biomarker, it requires some technical expertise, and it's a little bit laborious and time consuming to carry out. As opposed to in their system, they might be able to get results in a matter of hours, as opposed to, as they mentioned earlier, the total turnaround time traditionally is like 1 or 2 weeks, pretty much dramatically cutting down the turnaround time. So minimal non-specific sensor activation was observed in this heterogeneous population of IBD patients where diet and medications were not controlled, right? So, and they just took a largely random sample of patients. They didn't have them all on the same medication. They had different sort of varieties of IBD, so different disease manifestations. And even then, when it comes to multivitamin, there were different levels, or rather, some were taking them, some weren't. So yeah, as they say, heterogeneous patient population, and it still seems like their system worked just fine. So yeah, the medications were not controlled. So in addition, many of the patients, both with and without IBD, were taking daily multivitamin tablets, which usually contain 5 to 10 milligrams of zinc supplementation. Interestingly, this did not appear to affect the readout of our sensor as zinc is absorbed in the small bowel, which, yeah, that is an interesting finding because, again, going back to their system, they basically used genes that appear to be under the control of zinc. Like this addition of zinc wasn't interfering, which is, as they say, curious and interesting. But overall, our data suggests the feasibility of our engineered calprotectin sensing probiotic as a method of long-term, non-invasive monitoring of disease activity in IBD that would improve early detection of IBD relapses and clinical outcomes by reducing hospitalization. So again, pretty similar to this study here, if not not quite identical, almost as close to identical as you can get. In some sense, where, yeah, they engineered bacteria to detect tumor DNA, and like I said earlier, it was for the detection of colon cancer, or rather, tumor DNA from colon cancer. So they were still working in the GI tract, and again, as part of this study, they also looked, or rather, they also tested whether their bacteria basically was able to survive in the GI tract of mice. Basically carried out the exact same. So, several gut inflammation whole cell biosensors have been developed previously to detect nitrous oxide and tetrathionate as surrogate markers of gut inflammation. However, these targets were non-specific to IBD and have not been rigorously studied in the clinical setting as potential biomarkers for disease activity monitoring in IBD. So yeah, even though other research groups, they have come up with similar systems, okay, but they were not specific for these IBD patients. It was just like, and they weren't directly detecting inflammation. As they say, I think they mentioned, yeah, surrogate markers. Yeah, as surrogate markers of gut inflammation. So they're not directly detecting. Opposed to here, pretty much there is a direct correlation between calprotectin and the actual inflammation. Because again, the calprotectin, that's coming from the neutrophils, and the neutrophils are the type of white blood cell. So you have this large collection of white blood cells in the GI tract as a result of like the disease process that we don't fully understand. But that is contributing to the inflammatory process. But the neutrophils, they generate the calprotectin, and it's the calprotectin what is being detected here. So our model is geared to detect the current clinical gold standard of non-invasive disease activity monitoring for IBD, calprotectin, per the most recent clinical guidelines from the American College of Gastroenterology, and quantitatively tracks with the clinical laboratory results obtained from Northwestern Memorial Hospital Clinical Pathology Laboratory. Because, like, yeah, I have a bunch of reasons. Calprotectin. Okay, not a bunch, but quite a bit. Let's see. Yeah. So it's pretty, as they mentioned, gold standard. Oh, and Rafa's God, welcome. Good to see you. Is the clone version of an embryo considered artificial cloning? I don't know for sure, but my intuitions say probably, since though human cloning, I believe, is like banned worldwide, or like maybe there's some country that allow it. But I believe it's basically yeah, 70 countries had banned human cloning. So yeah, is the clone version of an embryo considered artificial in cloning? Sort of like maybe more of a philosophical question. But it's, yeah, still a very good question. I lean towards no, of like, I mean, if it's an actual clone, it's like, well, it would be still like the real thing. Like, it's just like a duplicate copy. But I mean, there's, I guess, the larger philosophical question of like, what is artificial? Where the dividing line is between artificial and not artificial, I don't think it's quite clear cut. But like, yeah, there's quite a bit of research showing that, this calprotectin is pretty much a really good marker or good way of tracking the inflammation that is occurring. And that, like, yeah, if through treatment or various interventions, you can get IBD patients where their calprotectin is low, as they say, it predicts sustained clinical remission in patients. Or in me too, because I am a patient. So in addition, so they're just highlighting that they're basically really trying to go by clinical guidelines of like the actual experts, the American College of Gastroenterology. It basically recommends that this calprotectin biomarker is tracked and that it's used to make medical clinical decisions for these patients. And that's why they're really focused on it. And that's why they're using it here to hopefully develop a better method. So in addition, the major advantages of our platform compared to conventional enzyme-based ELISA assays of calprotectin include simpler data analysis and on-demand assessment of samples, as it avoids the extraction step that is required for all ELISA-based assays. Specialized lab equipment is not required to develop our sensors or run the reactions as it is culture and plate reader based, right? So basically, the traditional methods, these ELISA methods, which are the gold standards, and ELISA has been around for a very long time. The enzyme-linked immunosorbent assays. This is like a mainstay for a lot of things, still used very widely in both medical settings and in research settings, even though there are clear drawbacks, and arguably, at least in my opinion, like there's much better methods than ELISA. But tradition is tradition, and it's very hard. Like once something is validated and becomes entrenched, very hard to get people to move away from it. This is not to say that it's useless or like bad. It's just like maybe, arguably, I think a little bit outdated. But yeah, all that to say, it's a pretty time-consuming, as they say, time-consuming test to carry out, and requires more processing and a little bit fancier equipment, and it actually requires more technical expertise. I would like to carry out an ELISA, which I have done. Not that it's super complicated, but let's say the skill of the person matters. How you manipulate things matters. Do you think that will ever change allowing human clones? Sorry. Oh, nothing to apologize for, Rafa. Well, I mean, I have AMA in my stream title, so I should live up to that. But it's a good question, and I've asked that question myself, to myself, where, human cloning, or even just cloning in general. Like, I don't know how I feel personally about people cloning their pets. Where, it would be great if human cloning, or rather, in some aspect, it would be great if human cloning was legal, and research could be carried out to, let's say, push the technology forward, because there's a whole bunch of applications that could be developed. Even just something like basically in the direction of like growing organs, right? Like if I can clone myself, okay, then I can hypothetically harvest my clone's organs, and whenever I want a new liver, I just get a genetically, I get my cloned liver, which no chance of rejection. I want a new heart. Again, pick your organ. So even just from that aspect, there's a lot of medical potential. But then there's I think, understandably, concerning side of like, basically, I don't think humanity can be trusted with this type of technology, at least not yet. Whether we'll actually get there, I think as of right now, it's banned for justifiable reasons, because I think it opens up a whole host of moral and ethical questions that I don't think we're ready, personally, to grapple with. Whether we'll actually be ready, I'm a little bit skeptical, and I don't actually know. So that is why, like, I agree with you, that can happen. Where it's like, when we clone someone, okay, we don't need to grow the whole organism, right? That's why it's like, we might be able to just clone, parts of me, right? You're not looking to clone my brain, not looking to clone everything about me. Maybe just, my liver, or like, most of me. But everything that makes me a person, so to speak, like, leave that out. That research on paper could be carried out and pushed forward. But unfortunately, there are bad actors out there that there's the concern that some nefarious researcher will not do that. And we saw this carried out with CRISPR. Where it's like, yeah, some researcher in China basically took things into their own hands, and they genetically modified, you know, some babies, hence the CRISPR baby. And it seems like this is just like a I don't know the entire stories and details, but from what I understand, like this is just like nefarious scientist who was a bad actor that did something that they shouldn't have done. And so I think there's the understandable concern when it comes to human cloning. Of like, yeah, on paper, okay, you can do ethical cloning, or you can do ethical research on cloning and push it forward, and not get to the point of like having to grapple with those moral and ethical questions. But unfortunately, there's just many bad actors out there in case of that. And so that's why I think it's understandably justified for human cloning to be currently banned. The actual technology for cloning is quite old. So, well, yeah, even then, that's why, what is actual cloning? But if I remember correctly, yeah, you are correct. Like, it's old, so to speak, or like, the preliminary research on cloning is old. And like, the first cloning endeavors that were, successful, that is old. Going back to like, yeah, 1990s. But from what I recall, like, yeah, that sheep, as you say, which was the most famous, but as highlighted, not the first animal to be cloned, I believe it was not like a perfect clone. I believe there was a whole host of, let's say, health issues that resulted. Yeah. Dolly was euthanized because she had a progressive lung disease and severe arthritis. Now, whether that's the result of the cloning process, I don't know. But if I remember correctly, but I could be misremembering, like basically, like the cloning, I think introduced a bunch of issues into whatever, and that yeah, a bunch of health issues resulted. That's why it's like, we can probably perfect it and make it and get it to the point of like cloning doesn't have any health risks. But yeah, the question is, can humans and scientists be trusted with that type of technology? I lean towards no right now. And even something like this, this artificial human embryo, there's a valid question to ask of like, can humans be trusted with pushing this forward? Which I think they are going to push this even more forward, right? Of like artificial embryos. Brave new world, as they say. So yeah, wrapping up. The incidence and prevalence of IBD have been increasing at a global level, particularly in developing and newly industrialized nations with historically low rates. In addition, accurate detection of relapsing disease is often delayed in these nations due to lack of reliable diagnostic facilities, particularly endoscopy and pathology. Our engineered calprotectin probiotic would play a key role in these patient populations as it offers a non-invasive, easy-to-use method of semi-quantitative determination of IBD disease activity that requires a simple fluorescent or luminescent readout. Offering accurate disease monitoring will result in appropriate risk stratification of patients and optimization of medical management as we strive to bridge the gap in healthcare disparity in these patient populations. Yeah, good summary. And basically, success, as in their engineered bacteria engineered for the purposes of detecting calprotectin to monitor inflammation in these patients. Successful. And that maybe opens the door for a variety of things, of like better patient monitoring, and also maybe better detection and diagnosis in developing countries. Because as they say in the closing paragraph, and as highlighted earlier, the prevalence and incidence of IBD appears to be increasing. And it seems to be increasing in developing countries, or rather countries that are essentially industrializing. And so it's like, well, yeah, there's a need for developing technology that can make it easier to detect and diagnose the condition, or at the very least, monitor the condition. Because as of right now, for detection and diagnosis, it requires pretty invasive medical testing. And even just like to monitor also often requires pretty invasive testing procedure. So basically this is moving in the direction of like, hey, we can monitor really well, maybe detect, probably not quite yet used for diagnosis, maybe probably never used for diagnosis. Yeah, probably not. But basically non-invasive surveillance in humans. Yeah, I think super cool. And again, mentioned over and over, there are multiple research groups, working sort of overall in the same direction of like engineering bacteria for a variety of purposes, not just detection of calprotectin, as I highlighted, for detecting tumor DNA, and I have other studies and I've come across other research of using engineered bacteria to detect a whole bunch of other things, not only just in humans and disease, but also even in the environment, etc., etc. Like, the world of synthetic biology, which is pretty cool world. So that's pretty much everything I wanted to cover. Yes. Don't need this. Yeah, I think that is actually everything that I want to cover. I wanted to basically mostly do a deep dive, on the paper, dive on, and covering the artificial embryo. Although, again, I didn't cover this entire paper. I'm probably never going to cover this entire paper outside Wheelhouse. But yeah, just covering the mainstream. It feels like we are so close to so many breakthroughs that could change the world. I wonder if people always felt like they were just going out on the brink of something great, being born too early. Yeah, totally. I agree with you wholeheartedly. Just like, yeah, the technological advancements that have occurred, and that based off of the research, what's in the pipeline of just like 5, 10 years, 15 years down the road, is pretty crazy to think about. And I feel pretty fortunate in terms of, where when I was born. But, I don't know any better. It's like, yeah, it's hard to say. And obviously impossible, so right now, to get out of your own subjective experience. Where it's like, yeah, I feel grateful for the time that I'm living in, personally. But whether my grandmother feels the same way, like, people older or younger than me, hard to say. But with that, I think I'm gonna, look for someone to raid. Because, yeah, I basically covered everything that I wanted to cover. Let's see who's streaming. Well, Astro Canuck is well. I mean, I'm just rating them over and over and over again. Set that up. But I want to thank everyone that passed by, and thank you so much, Rafa. Hope you have a lovely evening as well, and thank you so much for passing by. Thank you, non-smokers slash Struggle Session Radio for the raid and passing by. Super grateful. And yeah, shout out you. Very kind and generous. Not following Struggle Session Radio, you absolutely should be. And also shout out to Socrates for the raid. Also super kind and generous and fellow scientists. So all the more reason to follow them. And thank you, Doffy, for passing by. Thank you, Schlatsky. And thank you for the follow, Schlatsky. And thank you, Twin Tower Enjoyer and Struggle Session Radio for the follow. Super appreciated. Thank you, Bob Marley and Deflux Streams for coming in with the raid. And iCal Rich for passing by. Always great to see you. And DC Drunk, Bill Synthetic, and XROX Fist. Anybody else that I forgot, I apologize. But yeah, we're gonna raid into Astro Canuck. I plan to be back tomorrow around the same time. I might do maybe just a little bit of science and then some gaming. Probably just a lighter stream because today I actually had to prepare quite a bit. Because to do a deep dive on that paper, I want to make sure that I understood everything and sort of like plan out, figures that I want to talk about. So maybe as a break, maybe just tomorrow, maybe just some lighter science news, maybe some gaming, or just like reacting to, I don't know, we'll see. But I plan to be back tomorrow. And yeah, if you want to join the Discord, to ask questions, recommend stream topics, or just post science memes, by all means, more than welcome. Great resources and people in the Discord. And yeah, as usual, we're gonna raid into, I think for like the third night in a row, raid into Astro Canuck, Canadian astrophotography, science and tech streamer, and Lego streamer, and they have, again, a great stream setup. So yeah, with that, again, thank you everyone, super appreciated. And give a follow to Astro Canuck, although I'm sure I've rated them so often. Virtually everyone in my community following Astro Canuck. But yeah, until next time, take care, be well, be kind, and should be tomorrow.
This is what this whole stream was built upon. Camera.